In this video, we will discuss column chromatography, which is based upon specific molecular interactions, which mainly include non-covalent bonds. This approach is best for obtaining any biomolecule in its purest form, that is, if we have to purify a specific enzyme from a mixture of multiple cellular proteins. In this method, columns are filled with affinity beads consisting of metallic spacer arms and associated ligands that bind to the target compound. The type of the ligand used will depend on what we want to purify. For example, if we want to purify an enzyme, then ligand must be its substrate. And if we want to purify antibody, then our ligand must be an antigen, or to be more accurately, epitope. For sugar-based purification, we require sugar-binding agents known as lectins, and for biotin-based separation, we use avidin as ligands. Even there are various other techniques, for example, the diannexin 5 that recognizes phosphatidylserine. During apoptosis, the phosphatidylserines that are present on the inner side of the plasma membrane are exposed out. So, in order to differentiate between normal and apoptotic cell, we can use this technique. We can also use glutathione as transferase enzyme, and suppose we have created recombinant proteins and at the end we added 3 extra amino acids, glycine, cysteine and glycine, which is known as glutathione, and separating this recombinant protein from host protein can be done by passing all the cellular proteins from the column containing beads conjugated with glutathione as transferase. This allows purification of recombinant proteins from a mixture. Or we can also use polyhistidine tagging, then this can also be separated by nickel, which has a very high affinity for polyhistidine. So, these are some of the molecular recognition techniques which are used to obtain biomolecules in its purest form. So simply, we can say that when the heterogeneous mixture is loaded on the column, then most of the substance or impurities will be in the vas, but the target will remain bound with the beads functionalized with ligands. Now, how can we separate bound target substrate from the beads? This can be done by displacement method, in which we provide a free ligand in high concentration in the elution buffer. Other methods are also there, as we know that all non-covalent interactions are sensitive to pH. Due to change in this pH, there is change in the conformation of the biomolecules, which result in the dissociation of the target molecules from the beads. In other words, we can say that suppose we first used equilibrium buffer of pH 7 with heterogeneous mixture, and when it's time for elution, then changing the pH to say 9 or 5 so that there is change in the conformation of the biomolecules, and this will lead to alteration in interaction which were happening earlier between substrates and the functionalized beads. Suppose we have a polyhistidine tag protein, then at pH 7 it will remain in deprotonated form as its pKa is 6, so it will get associated with nickel. But when we lower the pH below 6, in that condition polyhistidine will carry positive charge, so it will now not interact with positively charged nickel. So we can say that we can use any type of method which has the potential to dissociate non-covalent interactions. So this is affinity chromatography, and when we compare it with other techniques, we can say that it is very specific technique, as we can only separate one target molecule at a time. Another type of chromatographic technique which we will discuss is hydrophobic chromatography, often referred to as reverse phase chromatography. In most of the chromatographic techniques we have discussed so far, the stationary phase used like paper, silica, gel beads, charge beads and affinity beads, all of them are polar, and generally we use non-polar mobile phase. So this is normal phase chromatography, that is, stationary phase is polar and mobile phase has non-polar character. But in case of reverse phase, the stationary phase is non-polar and the mobile phase is relatively polar, so This technique is known as reverse phase chromatography. In this technique, we have beads functionalized with aliphatic chains. Then a question arises. What will be the hydrophobicity of the column? The hydrophobicity will depend on the length of the R chain. That is, as we increase the length of R chain, then hydrophobic character will also increase. For example, when we compare C16, C18 and C20 columns, then C20 will be highly hydrophobic. This type of chromatographic technique is more preferred when we want to separate hydrophobic molecules. And if we use any of the previously discussed chromatographic techniques, then we will not be able to separate these compounds because in case of nonpolar molecules, there will be no difference in their polar property. So these compounds are unable to interact with the polar stationary phase. This can be seen in plant biotechnological application, where there are varieties of nonpolar molecules like terpenes, phenolics, alkaloid, cholesterol, and various secondary metabolites. Or in case of animal sciences like steroids, progesterone, and various steroidal hormones. And suppose in this column we have loaded 3 different samples. A is monoacyl glycerol, B is diacyl glycerol, and C is triacyl glycerol. And we have obtained a graph like this. Then what will be the peak for A, B, and C? Yes, the first peak is monoacyl glycerol, then diacyl glycerol, and triacyl glycerol. If we have used thin layer chromatography, then we will have obtained reverse sequence, because in case of reverse phase chromatography, hydrophobic compounds are more retarded and hydrophilic compounds will not get retarded as the stationary phase is hydrophobic. Now we will discuss another type of chromatographic technique, which is an automated version of column chromatography, which is known as high pressure liquid chromatography, HPLC. In this technique, we will get better resolution, as in this case the stationary phase is densely packed in column, which means that when solute molecules try to move through this column, it will experience more retardation force due to increased number of partition plates. Densely packed means the mobile phase will also not pass through the column freely. So we need to apply pressure. We apply pressure by using a pump. We can maintain flow rate like 1 ml per minute or 2 ml per minute, which depend on the pressure applied by the pump, that can vary from 1000 to 5000 psi. There is also a sample injection port so that solute samples can move forward along with the mobile phase. In this method, there is very less sample requirement, that is from 10 to 20 microliter, and that too at lower sample concentration ranging between 1 to 10 nanogram. But another chromatographic techniques require very large quantities of solute sample to form band and then get detected. We can also put some kind of detection system towards the elution site. Suppose we have some sort of light system at particular wavelength and we place a detector that collects the signal and send it to the processor, which process it and places it to the computer, and we get an absorbance versus time graph like this. Now, when only solvent passes through the detector, then we get absorption due to mobile phase only. We consider this absorption as baseline or zero absorbance, and when we inject a sample containing A, B and C, and suppose compound A reaches here and B and C are over here, the band which will move fast or slow will depend on the type of the column used, like porous beads, charged beads or any other type of column. So the principle of separation will depend on the type of the column that has been attached in the HPLC system. Now, as soon as the solute passes through the detection area, absorption will increase and then gradually decrease as the solute passes and get collected in different vials. After some time, we will get peak for B and C. So at the end, we are having the retention time of these compounds. And by matching the retention time of standard compounds, we are able to identify these compounds, as well as we can separate and quantify compounds based on the absorption value. More absorption value means more concentration and lower absorption means low concentration.